Thank you for joining our podcast, where I, Keely Severson, Eric Johnson, and Alicia Swami are exposing mold. Today we are here with Dr. Andrew Whitehead and two PhD students that are researching in his lab. We have Nicole McNabb, who is a PhD student of pharmacology and toxicology, and we have Tony Gill, who is a PhD student in genetics. Welcome, everybody. Thank you so much for joining us.
Thanks for having us.
So Alicia tells me that the topic that you all specialize in is evolutionary genomics. Who wants to tell us what that means?
I can take a stab at it. So we're interested in how species, mostly wildlife species, respond to changes in their environment, stressful changes in their environment, with a special focus on human-caused changes to the environment, like pollution and climate change. And species can respond physiologically over short time scales, or they can also change over very long time scales across many generations. And so we use genomics to understand mechanisms through which species could respond immediately, physiologically. So we try to understand the molecular responses, and that's where the genomics techniques and tools come in. Species can also respond over many generations through evolutionary change. And evolutionary change is associated with changes in the sequence of DNA. And that's where other genomic technologies come in, is looking at how DNA changes over time in response to changes in the environment, for example, through evolutionary adaptation. So we use genomics to understand physiological responses, mechanisms, and also genetic responses across longer periods of time.
So what things are you looking into right now in your research?
Well, I can talk a little bit about some of the killifish work that we're doing. And then Nicole and Tony perhaps can talk a bit about what they're doing in their research. So one long-term research theme in our lab is trying to understand how species respond to pollution, and in what circumstances species could evolve their way out of a scenario where they encounter pollution. And so we've been studying a really extraordinary example of that, where Atlantic killifish, they're a little fish about the size of your thumb, have very rapidly evolved pretty extraordinary resistance to really extreme levels of pollution that you find in some urban estuaries along the East Coast. And so these fish are normally very sensitive to pollutants, but populations that you find in urban estuaries have very rapidly evolved the ability to survive up to 8,000 times the normally lethal levels of chemicals, which is not subtle, and it's a massive evolutionary leap, and it's very unusual. Most species cannot or have not been able to evolve resistance to the pollution that we throw at them. So by studying killifish, these populations, we're trying to understand why adaptation, evolutionary adaptation, is so hard. By studying sort of the exception to the rule, we're gaining insight into why evolution is unlikely to be a solution and hasn't been a solution to pollution for most species. So Nicole is studying also killifish and their responses to pollution, a class of pollutants that's the public is only now is really starting to gain awareness of. And she's studying this really interesting time period of susceptibility. Nicole, do you want to tell them about your project a bit?
Sure, I can do that. So what Andrew was already talking about is thinking about it on a time scale of many generations, an evolutionary time scale. And traditionally, toxicology included looking at acute effects, like at an individual physiological level, so like within one individual's lifetime. But what my work involves is looking at multiple generations. So not quite on the other end of the time scale of many generations of evolution, but multiple generations. So like parent to children to grandchildren. And so I'm looking at exposures to flame retardants, which are ubiquitous. You can find levels of these flame retardants in humans and wildlife. And I'm looking at exposures in one generation of Atlantic killifish, and then looking at effects on their children and their grandchildren. So specifically, I'm looking at fish behavior, and that's partly because with these chemicals, there's evidence of neurological consequences and effects on behavior. And I'm also looking at other factors like reproduction and fertility and survival, of course, and then combining that with omics tools, so sequencing RNA and looking at what genes are turned off and on, and comparing that between generations, and comparing the unexposed to the exposed, or the exposed lineages. So like their grandparents were exposed to these flame retardants. And the benefit of using these omics technologies is that I can cast a broad net to look at some effects that we might not be measuring, because I'm not sure. Like the thing with these transgenerational effects is that we're not always sure what the effects will look like when they're passed along to future generations. So we can make a prediction, like I think maybe it will affect behavior, but not always sure what we might see. So that's the benefit of using these omics technologies.
Wow, thank you so much. And in terms of your transgenerational just overview, what are you guys finding? Are you at that stage where you're kind of finding some information and starting to put the pieces of the puzzle together?
Not quite, almost. So I just finished spawning the fish this summer to get the grandchildren. So this has been a study, now it's been over 2 years in the making since I first exposed the original generation of fish. So I'm not quite sure yet. So unfortunately, I don't have answers yet, but we did see some interesting effects in the behavior of the children. So the parents of the fish that I just spawned, but I'm not sure whether they're still persistent to the grandchildren.
This really reminds me of Dr. Shauna Swan's work with the PFAs in humans and how that's affecting our reproduction. And she's basically making the prediction that, every generation we're losing years and years of, sperm count and quality and all of that. So it's really interesting to see these environmental factors affecting nature, and also thinking about, and as I was discussing earlier, thinking about evolution, because we always think of evolution as something that happens slowly. But when you guys are doing this kind of research, and especially in the killifish, you're understanding that actually different factors can make a species evolve quite rapidly to their environment.
Yeah, I mean, that's been the prevailing attitude for a couple hundred years, right? That evolution and adaptation unfold slowly. I mean, certainly things like speciation unfolds slowly over long periods of time. So certain evolutionary processes are certainly slow, although there are some weird exceptions to that rule. But also that thinking has extended to adaptation. Even Darwin, when he presented his cogent theory of natural selection, still sort of considered adaptation as something that would unfold over very long periods of time. And that's true in many scenarios. But if you think of like the most obvious examples of natural selection and adaptation, we think of, pests evolving resistance to pesticides. We think of viruses evolving resistance to the chemicals that we throw at them. We think about tuberculosis evolving resistance to the drugs that we throw at them. So evolution can unfold very quickly, especially under sort of extreme environmental change. I think what a lot of people, there's this weird irony that I like to point out though, And that's the species that we want to kill tend to evolve resistance. So the insects and weeds that we want to wipe out from agricultural fields tend to evolve resistance to the chemicals that we throw at them. Diseases that plague us as people tend to evolve resistance to the chemicals that we throw at them. Yet the chemicals that we pollute the environment with, creatures that we care about preserving, fish, mammals, birds, don't evolve resistance. They tend to get wiped out. And so I think of that as a bit of an irony. And there are reasons for that. It tends to be a lot of the species that we want to kill are things that have features that enable them to evolve very, very, very quickly, like massive population sizes, very short generation times. So viruses, bacterial pathogens, insects, weeds, these are among the species that have massive reservoirs of genetic diversity, which is like the raw material for evolution. So they've got a lot of raw materials to evolve quickly, and they've got very short generation times, which means that they can pack in a lot of generations even just within a year. And they've got the capacity to make a ton of babies. So they have high reproductive potential. And those are all features of species that can enable them to evolve quickly. It turns out that the species, for better or worse, that we as humans tend to care about preserving, like frogs and birds and mammals and fish, are species compared to everything else that have pretty long generation times, have not that many babies relative to other species, and have relatively small genetic gas tanks. So have lower ability to evolve very quickly. So in the Anthropocene, in this current era of human impacts on the environment, we're seeing most species that we care about declining. We've all heard about the biodiversity crisis. And so the species that we care about are declining, and those that we have problems with are more likely to persist.
Thank you for that explanation. That was really, really great. And, as I was looking at your website online, I was really interested by the term ecotoxicology. Could you maybe discuss this a little bit more and what that means?
I could take a stab at that. Yeah. So I guess ecotoxicology, to kind of put it in perspective, is a kind of subfield of the larger of toxicology. And toxicology is basically fundamentally the study of study of poisons, right? And that's traditionally been done in looking at the effects of toxins and poisons in humans, right? And you can't obviously ethically poison humans to study what the effects are, but you can use animal models. So we've used animal models throughout history to study the effects of toxins, such as mouse models or fish models. Ecotoxicology, in contrast to that, is the study of toxins just in organisms in general, particularly in wild populations, right? So in wild populations, as opposed to contrast that with model organisms, you get kind of in the full range of variation in response to a contaminant in the environment. So whereas with inbred model organisms like mice, which are essentially, by inbred, they are essentially clones of one another. They have very little to zero genetic diversity, right? So by design, the full range of variation in response to a contaminant is restricted because you're basically working with clones, right? So focusing on wild populations and things that aren't model organisms and things that aren't just humans, expanding the range of toxicology to other organisms as well, you get it's more realistic in terms of understanding toxins in the environment, right? Their role that they play in the environment. However, this kind of embrace of variation, which I think is a good thing, does come with challenges, such as being able to determine the cause and effect, right? So let's say, for instance, when a population collapses in the wild, and it happens to coincide with a chemical spill in that same habitat, was it the spill that caused the collapse or decline in the population? Was it some combination of the spill and other unknown factors? In a controlled environment, a controlled laboratory environment, when you conduct chemical exposure studies, you control the environment completely, right? So you have a better grasp of cause and effect. You add a chemical and you see an effect in the organism, right? And so it's a challenge in ecotoxicology, but also I think it's a benefit is a kind of more realistic idea of what's going on. But like I said, there's establishing cause and effect can be a little more challenging. And I don't know if, and that's kind of where, I don't know if you want to talk about ecotoxicology too, establishing cause and effect and how we can kind of overcome that challenge and just some of the tools that we use in our lab to deal with that.
Right, I can jump in and add to it. So another big part of ecotoxicology is that we're either trying to hindcast or forecast, and I'll explain more what I mean by that. So with hindcasting, this is where something happened, there's a pollution event, say an oil spill, or we see significant negative effects in a wildlife population. So say there's a species of fish and we see populations declining, and we're trying to determine cause and effect of was that caused by exposure to pollution. And that can be really challenging, and you need strong evidence too, because say you're going up against a big oil company, if you're trying to say, well, your oil spill caused these fish to die, you need strong evidence to back that up. And so, one form to get that evidence would be to measure levels of the chemical in the animal, but that can be challenging because maybe it's a chemical that doesn't stick around in the body very long. So there are chemicals that are metabolized, broken down quickly and offloaded, so maybe we won't actually be able to measure that. But this is again where omics and molecular tools can come in handy, because we can actually look for known genetic signatures of exposure to that contaminant, and then combine that with, as Tony said, when we do experiments in the lab, we can control all of the variation and all the factors. And so if we combine field studies, so say we're going out and collecting organisms and sequencing their DNA to look for that molecular signature, combine that with an experiment in the lab where we expose the fish to the oil, and see whether there are negative health effects. So that's how we would build that strong evidence that the oil spill led to this negative effect of the fish population declining. So then on the other side with forecasting, this is where we're trying to predict possible health effects of chemicals. So there are thousands of chemicals, new chemicals coming onto the market every year. And a lot of times when they come onto the market, we don't know a lot about the potential health effects. So we might know that it's not toxic to the point of being lethal, but we don't know about potentially sublethal effects. So where it won't kill the animal, but could cause detrimental health effects such as reproduction. So with forecasting, a lot of times we're not really sure. We can predict what the effects might be, maybe based on the structure of the chemical, if it's similar to a structure of another chemical that's been well studied. So we can have predictions, but we're not really sure. And so again, that's where molecular tools help us cast a broad net for potential effects and actually generate hypotheses that we could further test with exposure experiments.
I remember about 20 years ago when the Delta smelt, the Sacramento Delta smelt started to crash. A sentinel species that was once the most numerous fish in the Sacramento Delta region, and the population just started to decline massively. And the working hypothesis was malnutrition, loss of habitat, warming water, just basically less places for the Delta smelt to do their thing. And a little bit of reading, and it turns out that they were suffering from severely deformed sex organs. They were, structural defects. So I contacted the California Department of Fish and Game and suggested that this was more consistent with endocrine receptor disruptors than with their working model. And they actually agreed and said that UC Davis was starting to work on that, look at the decline of the Delta smelt from that perspective. And yet in all the years since, they keep talking about loss of habitat and the other factors, and not the concept that this is some kind of ecological poisoning, nor the fact that this is actually in the drinking water for the Bay Area, which might possibly have effects on humans as well. Are you contacting people about possible effects in animal species, in fish, that could be extrapolated to the human population?
So scientists have been studying the Delta smelt decline for decades now. There's been a lot of resources spent on testing lots of different hypotheses. And it turns out that there are many causes. There isn't a single cause to Delta smelt decline. And people have been studying this for quite a while. As I mentioned, there's a very nice series of recent reports that I can send you, Eric, if you'd like to follow up. The answer is it's complicated because the changes that we have exerted on their native habitat are complicated. There are chemicals in the environment. There are radical changes to their habitat. There's changes to the flow. There's increasing temperatures. There's changes to predator-prey relationships. There's changes to introduced species that feed on them and that they feed on. There is no simple answer to why Delta smelt are declining. The simple answer is human alterations to the environment are what are declining. What are those human changes to the environment? Well, that's where establishing the chain of causality, establishing that chain of cause and effect that Nicole and Tony were talking about comes into play. And so we can pose hypotheses, endocrine disruptors, habitat loss, exporting of water, changes of salinity. But then we have to do the experiments and collect the data to fill in the gap between a hypothesis and an observation of population decline. And decades of research have shown that many of these factors interact. And some of those factors include chemical contaminants, like you raise, Eric. But that's not the only reason. Should we be concerned about chemicals in the environment? Yes. Should humans be concerned about chemicals in the environment? Do non-human species, are they sentinels for us? Yes. It's a lot, like Tony said at the beginning, we can't do experiments in people. We can collect epidemiological data to sort of make correlations between health effects in humans, so dropping sperm counts in men, and lots of things correlate with that. The causative factors are really hard to pull apart. But endocrine disruptors is probably a pretty good hypothesis. And we know that we are exposed to lots and lots and lots of chemicals. So like Nicole said, she works on flame retardants. Everyone on this phone call and everyone has detectable levels of flame retardants in their body. We have all been exposed and we have all accumulated these chemicals, and they're very persistent in the environment. They cause neurotoxicity, and they cause effects that will affect your children, even if your children weren't exposed. So should we be concerned about this? Yes. Is it hard to establish the chain of causality? Yes. But this is the role of science. And honestly, that's really the main challenge of ecotoxicology, like all science, is establishing this chain of causality. Anyone can propose a hypothesis, which imagines how one thing could cause another, but then the science has to be done to confirm that. And that's, I think, where a lot of people misunderstand, and some people even misrepresent science, right? Until causal relationships are established, hypothesis remains just a hypothesis. And when one keeps pushing that hypothesis as if causality has been determined when it hasn't is when, you know, that someone has hung up their lab coat, if they ever wore one, and becomes a person pushing an agenda. And we see a lot of this now, right? Especially with vaccines. A lot of people keep pushing these ideas, for example, that vaccines cause autism, or that vaccines cause Gulf War syndrome. But it doesn't take much digging to realize that there is no causality that has been established for these hypotheses, and not for lack of trying. So those that keep insisting on pushing these ideas as if they're true are pushing a pseudoscientific agenda. And so that's the real challenge here, is establishing this chain of causality. And I've gone off on a bit of a tangent, but to bring it back to delta smelt and a lot of the species that we study, it's complicated because we've wrought a lot of changes on the environment. And some of those changes, maybe there's one cause that really stands out, but a lot of these changes interact with each other in complex ways that takes a lot of hard science to figure out.
Yeah, one thing that I was curious about is whether or not the flame retardants might actually be processed by microbes into more pathogenic forms, that perhaps the flame retardants are not bioavailable and particularly harmful to humans in their purest form, but as they get into the environment, they can actually exacerbate microbial colonies and be processed into supertoxins.
For many chemicals, metabolic alterations tend to detoxify. There are examples where metabolic alterations will activate chemicals. That's what makes some pesticides toxic to insects and not humans, or at least reduce toxicity to humans, is because insects have the metabolic machinery to activate the parent compound into something that's toxic, whereas mammals don't. So a lot of designed poisons are designed in a way to not hurt people, but to hurt the target species. That doesn't always work perfectly, but that's the intent. And a lot of the chemicals that we're talking about here, like flame retardants, the reason flame retardants are a problem is because they're very tough. They don't get broken down in the environment. And that's why they're a problem. That's why chemicals that were banned 10, 20, 30 years ago are still out there in their parent form. And their parent form tends to be the most toxic form. And that's why they're a problem. If microbes could chew these up, we wouldn't be in the position we are. Chemicals like oil. So oil spills are clearly a big ecological problem. Some of the toxic components in oil we know are really toxic to humans, and they're in exhaust from cars, and a lot of the things that we breathe in. We're also experiencing essentially oil spills when we're breathing in exhaust from traffic. And these things can cause health impacts, but a lot of these chemicals get broken down very quickly by our bodies and by microbes. Does that mean they're not a problem? No, because we're persistently exposed to them, right? We keep getting hit with them. We keep metabolizing them, but not without damage happening in the meantime. Now, things like oil is a mixture of like 10,000 chemicals. Most of those chemicals, when they're attacked by microbes or by our own metabolic machinery, get broken down to less toxic forms. But some of them get metabolically activated to more toxic forms. That's the vast minority, but there are some hydrocarbons that become more cancer-causing with metabolic transformations. So yeah, that does happen sometimes.
Could there be a moment in time when the parent compound is being broken down by microbes and these materials are volatilized and become a toxic plume, airborne and more bioavailable? And yes, eventually they might be broken down into more harmless forms, but for that time when the microbes first start to decompose them, there could be a window of vulnerability when exposure to these microbial colonies could be more pathogenic.
I haven't seen evidence for that. There is a phenomenon called a global distillation, where chemicals that are of a certain structure that don't get broken down by microbes start to become a bigger problem. So chemicals that, there are lots of chemicals like PCBs that we've heard about, dioxins, and probably flame retardants, but I'm not sure, that in their parent form get volatilized. And as they get volatilized, they get moved by air currents away from where they were released into the environment. And especially in the summer, they volatilize as things warm up. They get up into the atmosphere and they move poleward, because that's how our air currents flow. They flow toward the poles, at least at higher elevations. And then later in the season, as it gets cold, that chemical can fall out and get back down to the earth. And the next summer, it volatilizes again and leapfrogs a little bit further poleward each year, further and further poleward. That's why people in northern climates, Inuit, polar bears, beluga whales, creatures that are farthest away from where pollutants are released in the environment, carry among the highest body burdens of these chemicals. So volatilization is a problem, and especially for chemicals that don't get easily broken down. And they can persist long enough to make it to an Arctic region and get concentrated in top predators.
Years ago, I had an experience where I was in an area that had been heavily firebombed or treated with borate from aerial drops of flame retardant to put out a forest fire. And I felt like I was having a strong reaction to this area. And I thought, is there any way without a laboratory, if I could find out if I'm reactive to this flame retardant? So I went to the loading platform at the airport and found that I had no problem with it. It didn't have any effect on me at all. But I went down to a culvert, a drainage ditch below the loading platform, and found that I was having a strong reaction down there. And that's what led me to the hypothesis that perhaps the microbes were breaking them down and releasing these substances, and that's what I was reacting to. And as it turns out, this area where this culvert was blowing, where the wind was directing this material downwind, everybody for about a mile was sick and complaining of headaches and fatigue and rashes and strange illnesses that no doctors could figure out. So that's why I was curious as to whether there could be a moment in time when the decomposition by microbes of flame retardants could release them and make them more bioavailable.
Yeah, I mean, so the kinds of observations that you mentioned there are important, right? This is where science starts. It starts with an observation. This is unusual. This isn't the way that it used to be. You know, this is, and you might notice an association. And that's the beginning of science, the formation of a hypothesis, and to seek mechanistic linkages between some event and some outcome, right? And so you've just now articulated some really good hypotheses, and you've made some associations. The science to follow up on that is the hard part, right? I think we can come up with lots of hypotheses, and where scientists go is they sort of pick the hypotheses that seem most plausible first, because we can have like lots of hypotheses, right? And start to chase those down and do experiments. I've not, and certainly microbes metabolize things, and metabolism can make things more or less bioavailable. And so there's certainly a hypothesis there. I haven't seen any evidence that starts to fill in that chain of causality, but the observation is a good place to start.
Yeah, the best I could do under the circumstance is lead other people who are chemically sensitive to these locations and ask them if they could feel anything. And it turns out they could. But of course, with no scientific abstracts backing up or supporting my claims, I can't get any researchers to come take a look at these places.
Sure. So we know that a lot of diseases are increasing in frequency, right, and have been for decades. Multiple cancers are increasing in frequency. Multiple neurological diseases have been increasing in frequency. And so why? That's the question. What are the causes of these things? And it's not because, and we know that there are different hotspots on the planet for a lot of disease. Okay, so certain autoimmune diseases you don't find in developing countries. You only find those in developing countries, in developed countries, sorry. And certain cancers, there are hotspots globally for certain cancers. And so what all this tells us is where there are hotspots and when there are things changing over time, that those are diseases that have environmental causes. So it's some change in the environment that is causing disease. So we think of, an individual's susceptibility to disease is a function of their genetics. Do you have mutations that might make you more susceptible to diabetes, for example, or mutations that might make you more susceptible to developing autism. And those exist, but those don't explain the change in disease over time. Because as a species, our genetics isn't changing over the time period where we've seen disease incidence increasing. So we know that susceptibility to disease is an interaction between our genetics and the environment. Environmental causes of disease and genetic causes of disease are both active areas of research. But for diseases that have been increasing in frequency recently, that's not genetics. That's something about the environment. So we can think of lots of reasons why lots of environmental changes, and chemicals is one of those things. We have been putting a lot more chemicals in the environment over the last century. And chemicals certainly contribute to human disease, and sometimes in very subtle ways, and sometimes in really obvious ways. And often that subtlety comes with low doses of exposures that don't cause an overt immediate effect, and mixtures of chemicals that might interact in very complex and in many ways unpredictable ways until you get that mixture through synergistic effects. And that makes them very hard to study.
Well, from an anecdotal, empirical, observational standpoint, at the same time, the Delta smelt and various other insect populations were crashing, the frogs simply disappeared. And the chytridiomycosis has been a terrible problem with some frog species going extinct. And I read about an amazing experiment, the Yellowleg frog up at Lake Tahoe. The populations crashed, and some researchers did a study where they took sick frogs with the chytrid disease up north, up beyond Mount Lassen, where the air is presumably cleaner because it's not downwind of the major metropolitan areas or the San Joaquin agricultural region. And some of the frogs that were clearly stressed and suffering with this fungal chytrid disease recovered. And they took healthy frogs from the Lassen area down to Lake Tahoe and introduced them into the same conditions, and they fell apart with this fungal problem. So there's an environmental correlation that was made here, very scary. Obviously, the Lake Tahoe Chamber of Commerce is not particularly interested in pursuing this. But here we have a study which shows that a low level of something in the environment, even at high elevations, is affecting the frogs. And to my knowledge, this has not been followed up on.
Yeah, there's lots of mysteries out there, which means that us as scientists aren't going to be without hypotheses to test. So that's some job security for us. And so, yeah, I mean, disease is very, very interesting. So, you know, this brings us to actually part of Tony's thesis research. So when in 1989, Exxon Valdez, you know, drunk captain runs an oil tanker aground in Prince William Sound in Alaska, and one of the most well-studied oil spills in history unfolds, you know, coating thousands of kilometers of pristine coastline in Alaska with oil. 3 years later, Prince William Sound herring collapses. 3 years later. It collapses, at least one of the major proximate causes was because a viral epidemic ripped through the population. Okay, so this has been a bit of a mystery. Not was there disease? Yes, there was disease, and yes, it was a problem for this population. But why then? This disease is present in other Alaskan populations and has never led to a full-on collapse. Was there a role for the oil spill 3 years before? And that's where Tony's research has taken off, because, you know, here diseases are also creatures that are behaving in an environment, and there can be changes to the environment that favor or disfavor them. So when we're thinking about climate change, you know, a lot of epidemiologists are very worried about disease being a relatively underappreciated threat to wildlife and humans, because as the temperature changes and as moisture patterns change, favorability and disfavorability of environments for disease changes. And so we're going to start seeing diseases in places that we didn't see them before. And we're going to start seeing diseases pop up in environments that were previously not favorable to them. And so diseases interact with the environment, and sometimes in complex ways. So yeah, this is something that we have some knowledge about, but we're gonna see some big surprises in the next few decades with disease outbreaks in wildlife and in humans.
Yeah, following up with what Andrew said, and kind of returning to our theme of establishing cause and effect, a little bit more about the Exxon Valdez oil spill. You know, the oil spill happened to coincide, it occurred in the spring, which is when herring are spawning. So you get this oil spill, happens during a spawn. So eggs, embryos, larvae are hatching, are growing up in early development, coated by oil, right? And however, the oil and the fish left that area and swam out into the ocean eventually. The oil also was either cleaned up or sunk to the bottom, where the fish were no longer in direct contact with it. And after about 2 years, there is very little detectable oil at the site of the spill, right? But as Andrew said, that the timing is different. So the collapse took place 3 to 4 years after the spill. So by the time the collapse happened, there was no more oil in the sound. So it's very hard to just make a direct connection between oil and collapse. There's an intermediate between there we have to establish. And that's kind of been, right, my thesis work here is trying to establish that, trying to connect those dots, if there are, you know, dots to connect, you know, if there is a line to be drawn. So yes, I'm interested in the effects of fossil fuels or hydrocarbons on your health. And so if you think particularly on the long-term impacts or long-term health consequences of exposure or coming into contact with hydrocarbons and fossil fuels. And we talked about this kind of before already, but long-term can kind of be defined in a couple different ways in our lab group. So for instance, if you come into contact with hydrocarbons or fossil fuels in early development, and for a kind of brief window of time early on in your development, early on in life, what are the long-term consequences of that? And then you're kind of like, you know, you're done being exposed to that. It was just for a brief window. Are there long-term consequences over the span of an organism's life, over the lifespan? Are there long-term consequences of that exposure to that contaminant? So that's one way to think about long-term. That's more like a developmental effect. And another way to think about it is are there long-term consequences over the generations, right? So your ancestors were exposed to a fossil fuel or had hydrocarbon exposure early on in their development. Does that echo through the generations and so that's more of on an evolutionary time scale. And so my work has been kind of looking at both of those in herring, both the developmental effects of brief early on exposure to hydrocarbons and fossil fuels, and then more of an evolutionary approach, looking at the long, over multiple generations. Is there still evidence? Is there still like a genomic imprint or genomic evidence or scar of this, of your ancestors being exposed to this? So it's pretty, I haven't published anything on it yet, but that's what we're trying to establish.
And of course, you must also be looking at intermediaries, the disruption in the food chain.
Mm-hmm. Right. My hypotheses moving forward are kind of the disease angle, more the pathogen angle. Does oil have some sort of immunotoxic effect on development? Does it affect the function of your immune system? Does this particular population who were exposed 30 years ago, are their ancestors, are their offspring many generations later still have an affected immune response to disease pathogens in the environment? But yeah, certainly there's a lot of contributing factors to the decline, to the collapse of that fishery, and it's also its lack of recovery too. And so ecosystem pressures are many and varied. Yeah, it's hard to tease that out. Yeah, but we're definitely.
So Eric, and that's a really good explanation, Tony. And Eric, you raise an important point. Fortunately, the Exxon Valdez spill has been probably the best studied oil spill in history. And what came from that was, a lot of ecotoxicology that really included a lot of ecology. And that's what ecotoxicology has honestly been lacking. There were a lot of head scratchers following the Exxon Valdez oil spill. And one was, why did river otters recover, but sea otters took a lot longer to recover? And that ended up being a food chain explanation. So it turns out that oil, even after it disappears from the surface, hasn't disappeared from the environment. And it can linger for a very long time in certain parts of the environment if you know where to look. So if you know where to go today, you could go to Alaska and you could dig up Exxon Valdez oil spill and kill a fish with it. So oil can persist for very long periods of time in the environment, especially in the sediments. And it turns out that both river otters and sea otters got whacked by the spill. River otters came back, sea otters didn't. Why? Because sea otters were digging for clams in the sediment. That's what they ate. River otters ate different things that weren't living in the sediment. So sea otters just kept feeding themselves creatures that were just sucking up oil continuously from the sediment. And so sea otters kept feeding themselves oil, essentially, through the food chain. And that was part of the explanation for their very, very slow recovery. So yeah, ecology matters. Things are interconnected. That's why it's very difficult to predict how species will respond to climate change. A lot of people have approached that by sort of asking which species are more or less resilient to high temperatures. Okay, that's something that we're doing actually in Delta smelt right now. Because you might imagine that species that can do just fine when the temperature is a few degrees higher might be one that you predict does well. But what if the species that species eats don't do well with increased temperatures? Even though you physiologically might be fine, if the species that you interact with in the ecosystem are not fine, then perhaps you're not going to be fine, not through direct effects of temperature on you, but through indirect effects through the ecosystem. And those are very hard to predict. So yeah, food chain things are definitely part of ecotoxicology, a big part of climate change biology. And that's where sort of complex systems modeling comes into play. Again, hard science to do, but science that is being done.
When I first started worrying about the delta smelt and these ecological changes that I was seeing, I read, of course, everything I could find looking for clues as to what was happening. And something that emerged was that boaters down in the delta were reporting green slime floating in the water that they could not identify. They saw this brightly colored green algae and would go to investigate. And sometimes just proximity, just in a boat, getting close to it, they would get headaches and nausea and severe neurological effects. And this was something that experienced boaters claimed that they had not seen before. So chasing these environmental changes up as far as you can, it looks like the occurrence of algal blooms, the harmful algal blooms that have become so prominent within the last 25 years. I don't know where they're coming from, the pollution, or what exactly creates them, or what makes them so toxic. But it seems that this goes along with the ecological changes that are affecting not only fish, but humans.
Huge research efforts by multiple institutes are investing in trying to understand the causes of harmful algal blooms, because you're right, they've been increasing in frequency. They're problematic for wildlife health and for human health, because we eat wildlife. They produce some harmful algal blooms are harmful because those creatures that make up the algal bloom produce toxic molecules, neurotoxins in many scenarios. And so neurological problems, neurological dysfunction, neurological toxicity is an outcome of exposures to those chemicals. Big problems for marine mammals. But yeah, what are the drivers? What are the environmental drivers of harmful algal blooms? Big area of research. I'm not an expert, so I'm not going to comment on it, but there are a lot of experts out there. Especially there's a big group at the Scripps Institute of Oceanography down in San Diego that have been big group down there that's been studying harmful algal blooms. Pretty much every oceanographic institute on the planet has someone there studying harmful algal blooms, because it's recognized to be a problem, at least partly driven by human-caused climate change.
Can you take zebrafish to different regions and observe them? Because they're a very sensitive species, very good for detection of these harmful pollutants. Are people doing this?
Yeah, I mean, people are using biosensors to understand as sentinels for environmental change. There is a program run by NOAA called Mussel Watch that you might have heard of. It's a number of sites along the various U.S. coastlines to use mussels, little bivalves, as sentinels for environmental health. Because mussels, filter a lot of seawater. So if there are chemicals in seawater, then mussels might accumulate them. And so again, that's getting at the question of bioavailability that you mentioned earlier, Eric. You can have chemicals in the environment, but if they're not being taken up by organisms, then they're going to be less of a problem than if they are taken up by organisms. So it's important to know what's being taken up by organisms. So let's look at the organisms. So you can either deploy organisms in the environment, for example, in cages, or you can just take advantage of organisms that are already there, like mussels, and go and sample them periodically and see if they're changing, if there's an increase or decrease in certain chemicals, if there are increases or decreases or changes in their reproductive physiology, increases or decreases in their survival and their ability to grow. So yes, biological sensors are in the environment, anywhere from people deploying cell systems that are molecular detectors to caging a fish, like a zebrafish or something else. You can't throw a zebrafish in the ocean though, so they'd be a freshwater sensor kind of species, to periodically sampling wild species. So this is actually something I did way back in my PhD research, is I was interested in how runoff from agricultural fields after spraying orchards was affecting creatures in the rivers. And this was done in the Sacramento-San Joaquin River system. And so what I did is I collected a native fish. So this was Sacramento sucker. And I collected them from an area that was clean, upstream of agriculture. And I kept them in a lab for many, many months. And then I basically put them in cages downstream of agricultural areas during rainstorm events, and basically sampled them during this rainstorm, this pesticide pulse event. And I basically assayed their biology. I used the fish to tell me, are you okay in this environment? And so that was one sort of example of deploying, a caged creature and assaying and examining its biology in response to an environmental change to establish that chain of causality. In the Deepwater Horizon oil spill, we did something a little bit different. Because we weren't prepared to deploy cages, because we didn't know a spill was going to happen, we basically went out and sampled wild fish from habitats where we saw the oil coming. So you remember that this oil spill was offshore, and so we had a few days to collect before pollution data, get a baseline of fish health along this coastline. And then one of those sites that we sampled from eventually got hit by oil. We could resample all of those field sites at that point and many times after. And through sort of space and time and the health of the animals, we were able to establish a chain of causality demonstrating the effect of that oil spill, on resident species. So that was an example where we did a field study, taking advantage of creatures that were already there to tell us something about the environment, to tell us something about their health and what were the causes. And then we've also done studies where we've bring creatures into the lab and do very controlled experiments to ask what we witnessed in the field, what we think is the cause, can we reproduce that in the lab and further build that strength of evidence to say this oil spill is causing developmental failure in this fish. And so lots of ways that you can get at that.
I wonder, so when you guys produce these research articles and you happen to find causality between, say, an oil spill and what's going on, is this information used to pursue action against the polluting agent? So that way they could at least pay for the ramifications or put money down or something into cleanup efforts or anything like that?
Yes. So, our research in particular was used to establish that the oil spill had damaged natural resources in the Gulf of Mexico, and was used by expert witnesses to establish that cause and effect relationship, and contributed to basically being able to establish that BP through and their subsidiaries through this accident and through their negligence had caused damages that they were on the hook for paying for. So yes, a lot of ecotoxicology research is really important for holding responsible parties accountable. So the stakes are high. And so the science has to be done well. It needs to, and again, that chain of causality is really important, because BP could say, oh, you detected reproductive failure in your fish. Not my fault, it wasn't the oil. It was something else along that coastline that caused that. How do you know that the reproductive failure in your fish was caused by our oil spill? but our lab studies, our field studies, our molecular tools that show a fingerprint of an oil response and not any other kind of response was crucial for building that pretty tight association, that pretty tight linkage, causal linkage between oil spill, developmental failure, that was really important. And it's not just us. We were one of many, many research groups that were able to establish chains of causality for lots of different damages to the environment caused by the oil spill. And as a group contributed to, a chain of causality that stood up in the court of law and held the responsible party accountable. A lot of the time, we just don't have the resources or the public backing to hold responsible parties accountable for the pollution that they cause. The science and the damages that cause. The science is out there for many of these kinds of cause and effect relationships. But the political, this is now becomes a political will and a public will to do something about it. Because banning chemicals that are problematic comes with economic costs. And there are powerful interests that are not interested in losing business and having to reinvent how they go about their business. And so big oil has known about climate change, has known about the burning of fossil fuels leading to climate change. They've known about this since the 1970s. They've known the cause-effect relationship. Some of the models that oil industry scientists built themselves in the 1980s fairly accurately predicted what's going on right now. And this is well documented. There are lots and lots and lots and lots and lots of research articles out there establishing this. The documents are available. But has that led to us as a people holding these businesses accountable for the environmental damage that they brought about? No. So it's now a political problem. The science is out there. So it's up to us, individuals, voters, people that care, to do something about it.
Absolutely. Thank you so much, Dr. Whitehead. And thank you all for coming on the show today. Eric Keeley, were there any last questions that you had for Dr. Whitehead's team?
Well, actually, yeah, I do have one final question. There was a tragic occurrence where a family died under mysterious circumstances. Some hikers out where harmful algal blooms were detected. And the cause of the mortality of this entire family and their dog was attributed to hyperthermia. And yet, I was wondering if it's possible that they got too close to this bloom and inhaled a little bit of antitoxin A, suffered enough neurological effects that this led to them possibly suffering from hyperthermia, whereas otherwise they would have been just fine.
Yeah, I know that story. I was aware of that happening at the time, and that was a terrible tragedy and a mystery for months. Because I have good colleagues that study harmful algal blooms and that work for excellent scientists that work for state agencies that are tracking the incidence of harmful algal blooms and some of the health effects that might be associated with. So that was a hypothesis early on, among many hypotheses of what caused this weird event of like 2 people and their dog. That's just bizarre. Foul play was ruled out early, is my understanding. And so that led to environmental causes. And my understanding is that the neurotoxic bloom was ruled out in favor. The evidence was more supportive of hyperthermia, heat stroke essentially. Whether there was some sort of interaction is, I guess, possible. Whether it's plausible is another question. I think, I could imagine a neurotoxin interacting perhaps with other neurological damages that could sort of compound each other and lead to maybe a synergistic event that can lead to mortality. But as far as I know, there aren't mechanisms that are understood where a neurotoxin could increase your susceptibility to a change in your thermal environment. But that's a hypothesis.
Very interesting. Thank you for asking that question and answering that question. Yeah, thank you again, everyone, for coming on the show today. I will share Tony's poster on the herring and also Dr. Whitehead, your lab's information as well. And actually, so we're wrapping up right now, and I'll be recording the closing. Now, if our audience members wanted to find any of you guys online, where can they do that?
Well, they can come to our website, and maybe, let's see, if you just Google my name, you will find UC Davis, you'll find our lab website. There's lots of information on the website about the research that we do and the research that we have published.
Fantastic. Sorry, I was getting ahead of myself for what I said earlier, but thank you again, Dr. Whitehead, and I'll go ahead and record the closing. Before we do that, was there anything else that you guys wanted to say or mention beyond where they can find you?
No, it's been a pleasure being on the show.
Yeah, thank you for having us.
Good, awesome. Thank you, guys. Thank you, everyone, for joining us today. It was a wonderful conversation with Dr. Andrew Whitehead and his team at UC Davis. They are studying evolutionary genomics. And the reason why I reached out to them is because I was really interested in understanding ecotoxicology and stress biology and what that means to fish and animals in the environment and how that can relate to humans. So we hope that you enjoyed this today. Please like, share, comment on our content. Also subscribe to our channel. We're on all podcasting platforms. And please go ahead and contribute to our GoFundMe and Patreon pages to keep this podcast rolling. Thank you, everyone, and we'll see you next time.